Hello there, and welcome to Whispered Knowledge. I'm so glad you found your way here tonight. Maybe you've had a long day filled with thoughts and decisions, or perhaps you're simply the kind of person who likes to drift off to sleep while exploring the wonders of science rather than counting sheep. Either way, you've come to exactly the right place. You've got nothing to do now but rest. Let your shoulders drop, your breathing slow, and your mind settle into a gentle rhythm. Because tonight, we're going to explore something truly extraordinary, something you carry with you every single moment of every single day, yet rarely stop to consider. Tonight, we're going to journey through the 3 pounds of tissue sitting between your ears, the human brain, a universe of electricity and chemistry, memory and mystery, folded into the space of a cantaloupe. Over the next while, you'll discover 100 facts about this remarkable organ. Some will surprise you. Others might explain experiences you've had, but never quite understood. We'll wander through neural pathways and chemical messengers, through the architecture of memory and the landscape of dreams. We'll visit the places where emotions live and where consciousness itself might arise. And we'll do it all slowly, gently, letting each discovery settle like snowflakes on a quiet winter night. So adjust your pillow, find that comfortable position you know so well, and let's begin this journey together. The brain has been waiting billions of years to tell you its secrets. The brain's basic architecture. The human brain weighs approximately 3 pounds. That's about the weight of a small bag of flour, or a half-gallon of milk sitting on your kitchen counter. Yet within those 3 pounds resides everything you've ever thought, felt, remembered, or dreamed. Every word you've spoken, every face you've recognized, every skill you've learned. 3 pounds. It seems impossible, doesn't it? That such a modest weight could contain an entire lifetime of experience. The brain doesn't feel heavy when you carry it around all day, moving through your world, because it's perfectly balanced within your skull, floating in cerebrospinal fluid like an astronaut suspended in the void. This fluid cushions it, protects it, keeps it from bumping against bone as you move through your day. 3 pounds of soft tissue containing more complexity than anything else we know of in the universe. The brain is the fattiest organ in your entire body. 60% of its weight comes from fat. Not the kind you might worry about on your waistline, but essential fatty acids that form the structure of every brain cell, every connection between neurons. These fats make up the myelin sheaths that wrap around nerve fibers like insulation around electrical wires, allowing signals to travel faster and more efficiently. Without these fats, your thoughts would move slowly, your reflexes would lag, your memories would struggle to form. The brain's dependence on fat reminds us that not all fats are bad. Some are absolutely essential. Some literally make thought possible. When you eat foods rich in omega-3 fatty acids, in fish and nuts and seeds, you're feeding your brain the very substance it needs to maintain itself, to keep rebuilding those cellular structures that allow consciousness to exist. Your brain contains approximately 86 billion neurons. Let me help you imagine that number. If you counted 1 neuron every second without stopping for sleep or food or rest, it would take you over 2,700 years to count them all. These neurons are the brain's primary workers, the cells that generate and transmit the electrical signals underlying every thought, every sensation, every movement. They come in many shapes and sizes. Some have long tendrils reaching across vast distances in the brain. Others stay close to home, making local connections. Each neuron is a tiny universe unto itself, with its own internal machinery, its own chemical factories, its own network of connections, reaching out to other neurons like citizens in an impossibly vast city, communicating constantly through the streets and pathways of your mind. Each of those 86 billion neurons can form connections with other neurons. We call these connections synapses. A single neuron might connect to thousands of other neurons, creating a web of almost incomprehensible complexity. Multiply this out across the entire brain, and you get a staggering number. Scientists estimate the human brain may contain somewhere in the range of 100 trillion synapses, though some research suggests it could be even higher. The exact number shifts constantly as you learn, as you forget, as your brain rewires itself in response to experience. These synapses are where the action happens. They're the gaps between neurons, the tiny spaces where one cell releases chemical messengers that drift across to the next cell, continuing the signal, passing along information like runners in an infinite relay race. Everything you are exists in the pattern of these connections, in which neurons talk to which, in how strongly they communicate, in the ever-shifting architecture of this vast neural network. The brain can be divided into several major structures. At the very back and bottom sits the brainstem, the oldest part of the brain in evolutionary terms, controlling basic life functions like breathing, heart rate, and sleep-wake cycles. Rising from the brainstem comes the cerebellum, that wrinkled ball of tissue at the back of your head, coordinating movement and balance with such precision that you can walk and talk and gesture all at the same time without thinking about it. Above these ancient structures blooms the cerebrum, the largest part of your brain, divided into two hemispheres like a walnut split down the middle. The cerebrum handles the functions we think of as distinctly human: language and reasoning, planning and creativity, conscious thought and voluntary movement. Its outer layer, the cerebral cortex, is only a few millimeters thick, but contains billions of neurons arranged in 6 layers, creating that characteristic wrinkled appearance. These wrinkles, these folds and grooves, serve a purpose. They increase surface area, packing more neural tissue into the limited space of your skull. The cerebrum divides further into 4 lobes on each side. At the front, the frontal lobes handle planning, decision-making, personality, and voluntary movement. They're what make you distinctly you, housing your ability to think about the future, to control impulses, to navigate social situations with grace or awkwardness. Just behind them sit the parietal lobes, processing sensory information from your body, helping you navigate space, understanding where your limbs are without having to look at them. At the back of your brain, the occipital lobes make sense of vision, transforming light patterns hitting your retinas into the rich visual world you experience. And tucked into the sides, above your ears, the temporal lobes process sound, language comprehension, and memory, working ceaselessly to make sense of the auditory world and to store the experiences that make up your autobiographical self. Deep beneath the cerebral cortex lie other crucial structures. The thalamus sits at the center of your brain like a relay station, receiving sensory information from the body and sending it to the appropriate regions of cortex for processing. Almost everything you sense passes through the thalamus first, except for smell, which has its own privileged pathway. Near the thalamus rests the hypothalamus, small but powerful, regulating hunger, thirst, body temperature, and the hormones that control everything from growth to stress, response to sexual function. The hypothalamus links your nervous system to your endocrine system, translating neural signals into chemical messages that flow through your bloodstream, coordinating body and brain into a unified whole. The brain consumes about 20% of your body's total energy, despite making up only about 2% of your body weight. Think about that for a moment. 1/50th of your body's mass uses 1/5 of its energy. That's a metabolic rate 10 times higher than the rest of your body's average. All those neurons firing, all those synapses releasing neurotransmitters, all that constant activity requires fuel. The brain runs primarily on glucose, burning through calories even when you're sitting perfectly still, thinking, remembering, dreaming. This enormous energy demand explains why you can feel mentally exhausted after a day of intense cognitive work, even if you never move from your chair. Your brain has been working the whole time, burning through resources, leaving you depleted. The brain generates enough electricity to power a small light bulb. About 20 watts of power flow through your neural networks at any given moment. This might not sound like much, but consider what it accomplishes. Every thought, every memory, every sensation results from patterns of electrical activity rippling through your neurons. These electrical signals move with incredible speed, traveling along nerve fibers at speeds up to 270 miles per hour. That's faster than any Formula One race car, faster than most bullet trains. When you touch something hot and jerk your hand away, that information travels from your fingertips to your spinal cord and back to your muscles in a fraction of a second. The brain's electrical system operates with a precision and efficiency that makes human-designed computers look clumsy by comparison. Your brain is wrinkled for a very good reason. Scientists estimate that if you could somehow smooth out all the folds and grooves of your cerebral cortex, it would cover an area somewhere between 2 and 2.5 square feet, roughly the size of a large pizza. But your skull is much smaller than that. The solution evolution found was to fold the cortex, creating those characteristic wrinkles we call gyri, and the grooves between them we call sulci. This folding allows far more cortical tissue to fit inside your skull than would be possible if the surface was smooth. More surface area means more neurons, more processing power, more capacity for complex thought. Other animals have brains too, but the human brain's particular pattern of folding, its particular degree of convolution, contributes to what makes human cognition distinctive. We can pack a remarkable amount of neural tissue into a relatively small space, all thanks to these wrinkles that look so strange but serve such an elegant purpose. The developing brain. The human brain grows at an astonishing rate during infancy. A newborn's brain is about 1/4 the size of an adult brain, but it triples in size during the first year of life. Think about that explosive growth. In just 12 months, the brain goes from about 350 grams to nearly 1,000 grams. This growth doesn't come from adding more neurons. Most of the neurons you'll ever have are present at birth. Instead, growth comes from neurons getting larger, developing more connections, and from glial cells multiplying. Glial cells support neurons in various ways, providing nutrition, cleaning up waste, insulating nerve fibers with myelin. During that first year, your brain was frantically building connections, laying down the neural infrastructure for everything you'd learn in the years ahead. Experience literally sculpts the developing brain. Every face a baby sees, every sound they hear, every touch they feel, strengthens certain synapses and weakens others, gradually building the neural networks that will support language, movement, memory, emotion. By age 3, a child's brain has formed approximately 1,000 trillion synaptic connections. Research suggests this is roughly twice as many as an adult brain maintains. This might seem backwards at first. Shouldn't adults have more connections, not fewer? But the developing brain follows a strategy of overproduction followed by pruning. In early childhood, the brain generates an excess of synapses, creating many more connections than it will ultimately need. Then, through a process called synaptic pruning, unused or weak connections are eliminated, while frequently used connections are strengthened. This pruning continues through childhood and adolescence, refining neural networks based on experience. The connections you use are kept and reinforced. The ones you don't use fade away. This is why early childhood experiences matter so much. The brain is at its most plastic, its most shapeable during these early years. What a child experiences, what they learn, what they practice, literally shapes the physical structure of their developing brain. The prefrontal cortex, that region at the front of your brain responsible for planning, impulse control, and decision-making, is one of the last brain regions to fully mature. It doesn't finish developing until around age 25. This delayed maturation of the prefrontal cortex explains a lot about adolescent behavior. Teenagers have fully developed limbic systems, the emotional centers of the brain, but their prefrontal cortex, which helps regulate those emotions and think through consequences, is still under construction. It's like having a powerful engine in a car with a still developing braking system. This mismatch can lead to impulsive decisions, risk-taking behavior, and difficulty with long-term planning. But it also contributes to the creativity, passion, and openness to experience that characterize adolescence. The brain's developmental timeline isn't a flaw, it's a feature, allowing for extended periods of learning and adaptation. Brain development proceeds from the back to the front. The brainstem and cerebellum mature first, ensuring that basic life functions and motor coordination develop early. Sensory cortices come next, allowing babies to make sense of the sights, sounds, and sensations flooding their world. The frontal lobes develop last, which is why higher order thinking, planning, and self-control emerge gradually across childhood and adolescence. This back-to-front progression makes sense from an evolutionary perspective. You need to breathe and move and sense the world before you need to plan for next Tuesday or contemplate abstract philosophical questions. The brain builds itself according to a logical sequence, establishing fundamental functions first, then gradually adding layers of complexity. Neuroplasticity, the brain's ability to reorganize itself by forming new neural connections, is most dramatic in childhood but continues throughout life. Your brain never stops being plastic, never stops being able to change in response to experience. When you learn a new skill, your brain physically changes. When you practice that skill repeatedly, those changes become more pronounced. Musicians who spend years practicing their instruments develop enlarged areas of motor cortex devoted to finger control. London taxi drivers, who must memorize thousands of streets and routes, have enlarged hippocampi, the brain regions involved in spatial memory. Your experiences literally sculpt your brain, strengthening some connections, weakening others, sometimes even generating entirely new neurons in certain brain regions. This plasticity is what makes learning possible at any age. Your brain at 60 can still form new memories, acquire new skills, adapt to new circumstances. It might take longer than it did when you were 7, but the fundamental capacity for change remains. The ability to learn language follows a critical period during early childhood. Young children soak up languages effortlessly, achieving native-like fluency in grammar and pronunciation almost automatically through mere exposure. But this window gradually closes. By puberty, learning a new language becomes much harder. Adults can certainly learn new languages, but they typically retain an accent and struggle more with grammar than children do. This critical period exists because the neural circuits supporting language are being established during early childhood. The brain is primed to extract linguistic patterns from the environment, to figure out which sounds matter in the language or languages it hears, to internalize the rules of grammar without explicit instruction. As these circuits mature and stabilize, they become less flexible, less able to accommodate entirely new linguistic systems. The brain optimizes itself for the language or languages of your environment, but in doing so, it sacrifices some of the flexibility it had in early childhood. Sleep plays a crucial role in brain development. During sleep, particularly during deep sleep, the brain consolidates memories and processes experiences from the day. For developing brains, sleep also provides time for synaptic pruning and neural reorganization. Babies and young children need far more sleep than adults, not because they're tired from physical activity, but because their brains are working overtime, processing the overwhelming amount of new information they encounter each day. When children don't get enough sleep, it can affect their mood, their ability to learn, their behavior, even their long-term brain development. Sleep is when the developing brain makes sense of its experiences, strengthening important connections and eliminating unnecessary ones. It's not wasted time. It's essential brain construction time. The brain's white matter, composed of myelinated nerve fibers that connect different brain regions, continues increasing into the 30s and 40s. Myelin is the fatty insulation that wraps around nerve fibers, allowing signals to travel faster and more efficiently. As we age through childhood, adolescence, and early adulthood, our brains continue adding myelin to nerve pathways, particularly those connecting distant brain regions. This myelination improves communication between brain areas, contributing to more integrated, more sophisticated thinking. It's one reason why cognitive abilities continue maturing well into adulthood. Long after neurons stop multiplying, the brain isn't just about the number of neurons you have. It's about how efficiently those neurons can communicate with each other. Myelination improves that efficiency, allowing for faster, more coordinated processing across the entire brain. Experience literally changes brain structure, not just in childhood, but throughout life. When adult rats are placed in enriched environments filled with toys, tunnels, and social interaction, their brains grow heavier, develop more synapses, and show increased production of new neurons in certain areas compared to rats in standard cages. The same principle applies to humans, living in a rich, stimulating environment, engaging in novel experiences, learning new things, maintaining social connections. All of these activities promote brain health and plasticity across the lifespan. Your brain responds to how you use it, strengthening the networks you engage most often. This is why staying mentally active matters, why learning new skills, pursuing hobbies, and maintaining curiosity all contribute to cognitive health as you age. Your brain is always listening to how you live your life, always adapting its structure to match your activities and experiences. The developing brain is particularly vulnerable to toxins and stress. Alcohol, drugs, lead exposure, chronic stress, all can interfere with normal brain development in ways that may have lasting consequences. The fetal brain is especially sensitive. This is why doctors strongly advise against alcohol consumption during pregnancy. Alcohol can disrupt neural migration, the process by which neurons move to their proper positions in the developing brain, leading to permanent structural abnormalities. Chronic stress in childhood can affect the developing limbic system, altering how the brain processes emotions and responds to threat, potentially increasing vulnerability to anxiety and depression later in life. The developing brain's plasticity, which allows for such remarkable learning and adaptation, also makes it vulnerable to disruption. What happens during development matters, shaping the brain's architecture in ways that can last a lifetime. Memory and learning. Memory isn't stored in any single location in your brain. Instead, memories are distributed across networks of neurons throughout the brain. When you remember your childhood home, Visual details might be stored in visual cortex. The emotional feeling of safety might involve the amygdala. The spatial layout might depend on the hippocampus, and the narrative of memories that happened there might engage frontal and temporal lobes. Remembering isn't like pulling a file from a cabinet. It's more like reassembling a jigsaw puzzle, with pieces scattered across different brain regions, all lighting up in a coordinated pattern. This distributed nature of memory means that brain damage rarely erases specific memories completely. Instead, damage might affect certain aspects of memory while leaving others intact. You might remember what your grandmother looked like, but not what she sounded like. You might remember events, but not the emotions attached to them. Memory is a symphony played by an orchestra scattered throughout your skull. The hippocampus plays a crucial role in forming new, long-term memories. This seahorse-shaped structure, deep in your temporal lobe, acts as a kind of memory consolidation system. When you experience something new, the hippocampus helps bind together the various sensory and cognitive elements of that experience into a coherent memory. Over time, usually over a period of days, weeks, or months, that memory gradually becomes independent of the hippocampus, taking up permanent residence in cortical networks. This process is called systems consolidation. The hippocampus remains essential for recalling recent memories, but older memories can often be retrieved even if the hippocampus is damaged. This is why people with hippocampal damage often have trouble forming new memories, but can still remember their distant past. The hippocampus is the gateway to long-term memory storage, but not the ultimate storage site itself. Working memory, your ability to hold information in mind for brief periods while working with it, has surprisingly limited capacity. Most people can hold about 4 items in working memory at once. Try remembering a phone number you just heard without writing it down. That's working memory in action. This limitation isn't a flaw. It's a feature that helps your brain focus on what matters most in the present moment. Working memory relies primarily on the prefrontal cortex, which maintains activity patterns representing the information you're holding in mind. This active maintenance requires metabolic energy, which is why mental work feels tiring. Working memory is the workspace of consciousness, the neural scratch pad where you manipulate ideas, solve problems, and make decisions. Its limited capacity means we can only think about a few things at once, forcing us to prioritize and focus. Long-term potentiation, often abbreviated as LTP, is one of the main cellular mechanisms underlying learning and memory. When 2 neurons fire together repeatedly, the connection between them strengthens. This is sometimes summarized as neurons that fire together, wire together. LTP involves changes at the synapse that make it easier for one neuron to activate the next. These changes can be temporary, or, with enough repetition, become permanent. This is the biological basis of practice and repetition. When you practice a skill over and over, you're inducing LTP in the neural circuits supporting that skill, making those circuits more efficient, more reliable. Eventually, the skill becomes automatic, requiring little conscious effort. The violinist's fingers find the right notes almost on their own. The basketball player's shooting motion becomes smooth and consistent. Your brain has physically changed, optimizing the neural pathways for a well-practiced action. The opposite of LTP also exists. Long-term depression, or LTD, is the weakening of synaptic connections. This might sound like a bad thing, but it's actually essential. LTD allows the brain to forget, to weaken unimportant connections, to make room for new learning. Without LTD, your brain would become cluttered with useless information, unable to distinguish between what matters and what doesn't. The balance between LTP and LTD shapes neural networks, strengthening useful connections while pruning away those that serve no purpose. This constant remodeling continues throughout life, allowing your brain to adapt to changing circumstances, to learn from experience, to optimize itself for the environment and challenges you face. Memories are not perfect recordings of past events. Every time you recall a memory, you reconstruct it, and in the process of reconstruction, you can unintentionally modify it. Details might be added, subtracted, or changed. Emotions associated with the memory might intensify or fade. Over many retrievals, a memory can drift significantly from the original event. This is why eyewitness testimony, though compelling, can be unreliable. People remember what they think happened, but their recollection has been shaped by subsequent experiences, by things they were told, by their own expectations and beliefs. The brain doesn't work like a video camera, faithfully recording everything. It works more like a storyteller, reconstructing narratives from fragments, filling in gaps with plausible details, creating coherent stories from incomplete information. This reconstructive nature of memory makes us all unreliable narrators of our own past, but it also makes memory flexible and creative, able to extract meaning and patterns rather than simply storing raw data. Emotional events tend to be remembered more vividly than neutral events. This makes evolutionary sense. Emotionally significant events, particularly those involving danger, reward, or social connection, are worth remembering. They contain information that might be important for survival and reproduction. The amygdala, a key structure in emotional processing, modulates memory formation in other brain regions, particularly the hippocampus. When something emotionally arousing happens, the amygdala signals to the hippocampus and other memory structures, essentially saying, "This is important, remember this." The result is stronger, more detailed memories. This is why you probably remember where you were during major life events or significant historical moments, but have no memory of ordinary Tuesday afternoons from 5 years ago. The brain prioritizes emotional memories because they matter more, because they're more likely to contain information relevant to your survival and well-being. Sleep plays a critical role in memory consolidation. During sleep, particularly during slow-wave sleep and REM sleep, the brain replays neural activity patterns from waking experience, strengthening important memories and integrating them into existing knowledge. Sleep deprivation impairs memory formation, making it harder to learn new information and to remember what you've learned. Students who pull all-nighters before exams often perform worse than those who study less, but get adequate sleep. This happens because sleep is when memories move from temporary storage in the hippocampus to more permanent storage in the cortex. Without sleep, this transfer process is disrupted, leaving memories fragile and prone to forgetting. Sleep isn't downtime for the brain. It's active processing time, essential for turning experiences into lasting memories. The act of remembering, of retrieving a memory, actually strengthens that memory. This is called the testing effect, and it has important implications for learning. Reading material over and over has limited benefit, but actively trying to recall that material from memory, even if you struggle, strengthens the memory much more effectively. This is why practice tests, flashcards, and trying to explain concepts in your own words works so well for learning. The retrieval process itself causes changes at synapses, making those memories easier to access in the future. Every time you successfully remember something, you make it more likely that you'll remember it again. Memory is self-reinforcing. The more you use it, the stronger it becomes. This retrieval practice principle has profound implications for education and learning. Students who spend time retrieving information from memory rather than simply rereading material learn more effectively and retain information longer. The struggle of trying to remember something, even when it's difficult, creates learning. This seems counterintuitive because retrieval practice often feels harder and less productive than rereading. When you reread material, it feels familiar, and this familiarity creates an illusion of learning. But familiarity isn't the same as actually remembering. When you test yourself, when you struggle to retrieve information, you're strengthening the neural pathways that will allow you to access that information in the future. The difficulty is the point. It's what drives learning. Spaced repetition, reviewing material at increasing intervals over time, is another powerful learning technique based on how memory works. When you review something just as you're about to forget it, you strengthen the memory more effectively than reviewing it when it's still fresh. Spacing out your practice sessions, rather than cramming everything into one session, leads to better long-term retention. This happens because each retrieval, especially when it requires effort, strengthens the memory trace. If you review something when it's still very accessible, the retrieval is easy but doesn't create much additional strengthening. If you wait until the memory has started to fade, the retrieval requires more effort and creates more strengthening. Optimal spacing means reviewing material when it's challenging, but still possible to retrieve, maximizing the learning benefit of each review session. Forgetting isn't always a failure of memory. Sometimes it's an adaptive feature. Your brain is constantly bombarded with information, most of which is irrelevant. If you remembered every single thing you experienced, your memory would be cluttered with useless details, making it harder to access important information. Forgetting allows your brain to prioritize, to keep what matters, and discard what doesn't. There are even rare individuals with highly superior autobiographical memory, people who can remember nearly every day of their lives in extraordinary detail. While this might sound like a gift, many report that it's actually burdensome. They struggle to forget painful experiences, to let go of grudges, to move past embarrassing moments. Their memories remain fresh and vivid, long after most people would have forgotten. The ability to forget, to let memories fade, turns out to be as important as the ability to remember. Emotions and the limbic system. The limbic system, a collection of interconnected brain structures, plays a central role in emotion, motivation, and memory. This system includes the amygdala, hippocampus, hypothalamus, singular gyrus, and several other structures. The term limbic comes from the Latin word for border, referring to the fact that these structures form a border around the brainstem, sitting between the older, more primitive parts of the brain and the newer cerebral cortex. The limbic system represents an intermediate level of brain evolution, adding emotional and motivational complexity to basic survival functions. It's what gives mammals their distinctive mammalian qualities: the ability to form social bonds, to experience complex emotions, to be motivated by more than just immediate biological needs. The amygdala, an almond-shaped structure in each temporal lobe, is often called the brain's fear center, though that's an oversimplification. The amygdala certainly plays a crucial role in processing fear and detecting threats, but it's involved in other emotions too, including pleasure, anger, and anxiety. When you encounter something potentially dangerous, your amygdala can trigger a cascade of responses even before you're consciously aware of the threat. Your heart rate increases, your breathing quickens. Stress hormones flood your bloodstream, preparing you for fight or flight. This rapid response system helped our ancestors survive, allowing them to react to predators before taking time to consciously think about the danger. In modern life, the amygdala can sometimes be overactive, responding to social threats or imagined dangers as if they were life-threatening. This contributes to anxiety disorders, where the brain's alarm system becomes too sensitive, triggering fear responses in situations that don't warrant them. The amygdala also helps form emotional memories. When something emotionally significant happens, the amygdala modulates memory formation in other brain regions, particularly the hippocampus. This is why emotional events, especially fearful ones, create strong memories. The amygdala essentially tags certain experiences as important, ensuring they're remembered. This system usually serves you well, helping you remember dangers to avoid, but it can also contribute to psychological problems. In post-traumatic stress disorder, the amygdala becomes hyper-responsive, causing intrusive memories of traumatic events and triggering fear responses to reminders of the trauma. The amygdala has learned too well, creating memories that are too vivid, too persistent, too easily triggered. The hippocampus, sitting right next to the amygdala, serves as the brain's memory center, particularly for declarative memories, the kind of memories you can consciously recall and describe. It plays a crucial role in spatial memory, helping you navigate through your environment and remember where things are. Studies of London taxi drivers found that their hippocampi are larger than average, particularly the posterior region, likely enlarged by years of learning and remembering thousands of routes through the city streets. The hippocampus is one of the few brain regions where new neurons continue to be born in adulthood, a process called adult neurogenesis. Exercise, learning, and environmental enrichment all promote neurogenesis in the hippocampus, which may contribute to memory improvement and mood regulation. Damage to the hippocampus can result in profound memory problems. The most famous case is that of Henry Molaison, known in the scientific literature as patient HM, who had large portions of both hippocampi surgically removed to treat severe epilepsy. After the surgery, he could no longer form new long-term memories. He could remember his life before the surgery, could carry on a conversation, could learn new motor skills, but every new experience vanished from his memory within minutes. He would read the same magazine over and over, always finding it fresh. People he met post-surgery remained strangers no matter how many times they met. HM's tragic case revealed just how essential the hippocampus is for creating new memories, while also showing that many types of memory and learning don't depend on the hippocampus at all. He could learn new motor skills, even though he couldn't remember learning them. His case transformed our understanding of memory, showing that different types of memory involve different brain systems. The hypothalamus, despite being about the size of an almond, controls an astonishing range of functions. It regulates body temperature, hunger, thirst, fatigue, sleep, and circadian rhythms. It controls the release of hormones from the pituitary gland, linking the nervous system to the endocrine system. When you feel hungry, that sensation originates partly in the hypothalamus. When you feel thirsty, the hypothalamus is involved. When you're attracted to someone, the hypothalamus plays a role in that too, triggering the release of hormones that influence sexual behavior. This tiny structure serves as a control center for homeostasis, constantly monitoring and adjusting various body parameters to keep everything in balance. It receives information about your body's internal state and responds by initiating appropriate behavioral and physiological changes. The cingulate cortex, a curved band of cortex running along the midline of each cerebral hemisphere, connects the limbic system with the prefrontal cortex, integrating emotion with cognition. The anterior cingulate cortex, the front portion, appears to be involved in error detection, conflict monitoring, and decision-making. It becomes active when you have to choose between competing options or when you make a mistake. Some researchers think it helps signal when you need to pay more attention or adjust your behavior. The posterior cingulate cortex connects to the hippocampus and appears to be involved in memory retrieval and spatial orientation. Damage to the cingulate cortex can result in emotional disturbances, including apathy, a lack of emotional response that can be as debilitating as excessive emotional reactivity. Your brain processes different emotions in different ways and in different regions. While the amygdala is crucial for fear, other emotions engage different networks. The insula, a region of cortex hidden deep within the lateral fissure, separating the temporal and frontal lobes, processes disgust. When you see something revolting, your insula activates. Interestingly, the insula also activates when you experience social rejection or witness unfair treatment. Disgust, it seems, isn't just about contaminated food or unpleasant smells. It extends to social and moral domains as well. The brain uses some of the same neural machinery to process physical disgust and moral disgust, which might explain why we use the same word for both and describe morally objectionable behavior as disgusting. Happiness and pleasure involve activation of reward circuits in the brain, particularly structures like the nucleus accumbens and the ventral tegmental area. These regions are rich in dopamine neurons, and dopamine release in these circuits creates feelings of pleasure and motivation. This reward system evolved to reinforce behaviors important for survival and reproduction, like eating, drinking, and sex. But it can be hijacked by drugs, by gambling, by any activity that produces intense pleasure. Understanding how the brain's reward system works helps explain addiction. Drugs of abuse often produce massive dopamine release, overwhelming the system, creating pleasure far more intense than any natural reward. Over time, the brain adapts, becoming less sensitive, requiring more of the drug to achieve the same effect, driving the compulsive drug-seeking behavior characteristic of addiction. Emotions aren't just mental experiences, they involve your entire body, coordinated by brain structures like the hypothalamus and amygdala. When you feel fear, your heart pounds, your muscles tense, your palms sweat, your digestion slows. These bodily changes are part of the emotion, not just side effects. The philosopher William James proposed over a century ago that emotions are actually the brain's perception of bodily changes. You don't run because you're afraid. You're afraid because you notice yourself running, heart pounding, breath quick. Modern research suggests the relationship between body and emotion is more complex than James proposed, but he was right that emotions involve the body intimately. The brain constantly monitors signals from your heart, lungs, gut, muscles, and other organs, and these bodily signals influence emotional experience. This is why physical interventions, like deep breathing, progressive muscle relaxation, or exercise, can affect your mood. They change the signals your brain receives from your body, which in turn changes emotional experience. Chemical messengers. Neurotransmitters are the chemical messengers that allow neurons to communicate across synapses. When an electrical signal reaches the end of a neuron, it triggers the release of neurotransmitters, molecules that float across the tiny synaptic gap to the next neuron, where they bind to receptors and influence whether that next neuron will fire. More than 100 different neurotransmitters have been identified, each with its own specific functions and effects. Some neurotransmitters are excitatory, making the next neuron more likely to fire. Others are inhibitory, making it less likely to fire. Many neurotransmitters also act as neuromodulators, subtly adjusting the sensitivity of neural circuits, changing how responsive neurons are to other signals. The brain's chemistry is complex, with different neurotransmitters working in concert, sometimes reinforcing each other's effects, sometimes opposing them, creating the intricate dance of neural activity underlying every thought, sensation, and action. Dopamine is perhaps the most famous neurotransmitter, often called the brain's pleasure chemical, though that's not quite accurate. Dopamine is more involved in motivation and reward anticipation than in pleasure itself. When you expect a reward, when you're working toward a goal, dopamine levels rise, driving motivated behavior. The actual receipt of the reward triggers dopamine release too, but anticipation is key. This is why dopamine plays such a central role in addiction. Drugs of abuse cause massive dopamine release, hijacking the reward system, making the brain believe that the drug is immensely valuable, more important than food, water, or social connection. But dopamine isn't just about addiction. It's crucial for normal motivation, for the desire to pursue goals, to seek out rewards, to engage with the world. Low dopamine is implicated in depression, where nothing feels rewarding, where motivation evaporates. It's also central to Parkinson's disease, where loss of dopamine-producing neurons causes movement difficulties. Serotonin influences mood, sleep, appetite, and social behavior. Most of your body's serotonin is actually in your gut, where it helps regulate digestion. But the serotonin in your brain plays a very different role. It's involved in regulating mood, with low serotonin levels associated with depression. This is why many antidepressant medications, called selective serotonin reuptake inhibitors, or SSRIs, work by increasing serotonin levels in the brain. They block the reuptake of serotonin from the synapse, leaving more serotonin available to bind to receptors on the receiving neuron. Serotonin also influences sleep, particularly the transition between waking and sleeping, and the regulation of REM sleep. It affects appetite, with higher serotonin levels generally reducing food intake. And research suggests it may play a role in social behavior, with some studies indicating that serotonin could influence social decision-making, trust, and cooperation. GABA, short for gamma-aminobutyric acid, is the brain's main inhibitory neurotransmitter. While that might sound negative, inhibition is absolutely crucial for proper brain function. Without GABA, neural activity would run wild, spreading uncontrollably through neural networks. GABA puts the brakes on neural firing, preventing excessive excitation, maintaining balance. Many anti-anxiety medications work by enhancing GABA's effects, calming overactive neural circuits. Alcohol also enhances GABA activity, which contributes to its relaxing effects at low doses and its sedating effects at higher doses. Too little GABA activity can result in anxiety, insomnia, and even seizures. The brain requires a careful balance between excitation and inhibition, between signals that say go and signals that say stop. GABA is essential to that balance. Glutamate is the brain's main excitatory neurotransmitter, complementing GABA's inhibitory role. Most synapses in the brain use glutamate. It drives neural activity, passing signals forward, allowing information to flow through neural networks. Glutamate is crucial for learning and memory, particularly for long-term potentiation, the synaptic strengthening that underlies memory formation. But too much glutamate can be dangerous. Excessive glutamate release, such as occurs during a stroke, when oxygen-deprived neurons dump their glutamate all at once, can overstimulate neurons to death, a process called excitotoxicity. This is one reason why strokes cause brain damage. The initial loss of oxygen is bad enough, but the flood of glutamate that follows makes things worse, killing neurons that might otherwise have recovered. Acetylcholine was the first neurotransmitter ever discovered, identified in the early 20th century. It plays crucial roles both in the brain and in the peripheral nervous system. In the body, acetylcholine activates muscles, causing them to contract. In the brain, it's involved in attention, learning, and memory. The loss of acetylcholine-producing neurons in the basal forebrain is one of the hallmarks of Alzheimer's disease, contributing to the memory problems and cognitive decline characteristic of the disease. Some Alzheimer's medications work by inhibiting the breakdown of acetylcholine, increasing levels in the brain, which can temporarily slow cognitive decline. Acetylcholine also plays a role in REM sleep, with acetylcholine levels rising during REM, contributing to the vivid dreams and brain activity characteristic of this sleep stage. Norepinephrine, also called noradrenaline, is involved in arousal, attention, and the stress response. It's one of the neurotransmitters mediating your fight or flight response. When you're startled or stressed, norepinephrine levels spike, increasing heart rate, sharpening attention, preparing you to respond to danger. This same system, when dysregulated, can contribute to anxiety disorders. ADHD, attention deficit hyperactivity disorder, involves problems with norepinephrine signaling in prefrontal cortex, contributing to difficulties with attention and impulse control. Medications used to treat ADHD often work by increasing norepinephrine levels in the brain, improving attention and reducing impulsivity. Endorphins are the brain's natural opioids, producing pain relief and feelings of pleasure. The word endorphin literally means endogenous morphine, morphine produced within the body. Endorphins are released during exercise, and research suggests they may contribute to the phenomenon known as runner's high, though scientists continue to study exactly which chemicals create this effect. They're also released during laughter, during sex, and when eating certain foods. Endorphins bind to the same receptors in the brain as opioid drugs like morphine and heroin, producing similar effects, though typically less intense. Understanding endorphins helps explain how opioid drugs work and why they're so addictive. They hijack a natural reward system, producing pleasure far more intense than any normal activity, gradually altering brain chemistry and creating dependence. Oxytocin is sometimes called the love hormone or the bonding hormone because of its role in social attachment. It's released during childbirth, helping trigger contractions and stimulating milk production. It's also released during breastfeeding, promoting bonding between mother and infant. Oxytocin is released during sexual activity, particularly during orgasm, which may promote pair bonding. It's also released during positive social interactions, during hugging, during moments of trust and connection. Research suggests oxytocin may promote social behavior, trust, and empathy. Studies have shown that giving people oxytocin in a laboratory setting can influence their willingness to trust others, to cooperate, and to read emotions from facial expressions. But oxytocin's effects are more complex than simply making people friendly. Some research indicates it can also enhance in-group loyalty, sometimes at the expense of outgroups. The brain's chemistry of social behavior turns out to be nuanced, context-dependent, far more subtle than simple models of good and bad chemicals might suggest. The balance between different neurotransmitters is often more important than absolute levels of any single neurotransmitter. Mental health conditions often involve imbalances, with some systems overactive and others underactive. Depression might involve low serotonin, but also problems with dopamine and norepinephrine. Anxiety might involve problems with GABA inhibition and excessive norepinephrine. Schizophrenia is associated with excessive dopamine activity in some brain regions, but reduced activity in others. This complexity explains why treating mental health conditions is challenging. A medication that boosts one neurotransmitter might help some symptoms but make others worse. Finding the right treatment often requires trial and error, adjusting medications and dosages to find the right balance for that particular individual's brain chemistry, sleep, dreams, and consciousness. Sleep is one of the great mysteries of neuroscience. Every animal with a nervous system sleeps or engages in something sleep-like. This universality suggests that sleep serves some fundamental, essential function. Yet, for a long time, scientists couldn't identify exactly what that function was. We now know that sleep serves multiple purposes. It's essential for memory consolidation, for processing emotions, for clearing metabolic waste from the brain, for maintaining immune function, for regulating metabolism and appetite. Sleep is when the brain reviews the day's experiences, deciding what to remember and what to forget, integrating new information into existing knowledge, making connections between seemingly unrelated ideas. Sleep is active processing time, essential for proper brain function. Your brain cycles through different stages of sleep throughout the night. When you first fall asleep, you enter non-REM sleep, which progresses through 3 stages, each deeper than the last. Stage 1 is light sleep, a transitional state between waking and sleeping. Stage 2 is slightly deeper, characterized by sleep spindles and K-complexes, patterns of brain activity that may play a role in memory consolidation. Stage 3 is deep sleep, also called slow wave sleep because of the large, slow brain waves that dominate the EEG recording during this stage. This is the deepest, most restorative sleep. It's very hard to wake someone from stage 3 sleep, and if you do manage it, they'll likely be confused and groggy. Stage 3 sleep is essential for physical restoration, for immune function, for feeling refreshed and alert the next day. After cycling through the stages of non-REM sleep, you enter REM sleep, named for the rapid eye movements that occur during this stage. REM sleep is paradoxical. Your brain becomes highly active, almost as active as when you're awake, but your body becomes paralyzed, unable to move except for your eyes and diaphragm. This paralysis protects you from acting out your dreams. When the system that induces this paralysis malfunctions, people can physically enact their dreams, sometimes injuring themselves or their bed partners. REM sleep is when most vivid dreaming occurs. The content of REM dreams tends to be bizarre, emotional, hallucinatory, featuring impossible scenarios and illogical narratives. This bizarreness might reflect reduced activity in the prefrontal cortex during REM sleep, the same brain region responsible for logical thinking and reality testing when you're awake. You cycle between non-REM and REM sleep multiple times each night, with each cycle lasting about 90 minutes. Early in the night, non-REM sleep dominates, particularly stage 3 deep sleep. As the night progresses, REM periods become longer and deep sleep becomes less prominent. By morning, most of your sleep consists of stage 2, non-REM, and REM sleep. This architecture of sleep serves different purposes at different times. Deep sleep early in the night allows for physical restoration and memory consolidation. REM sleep later in the night may help with emotional processing and creative problem solving. If you're sleep deprived and then catch up on sleep, your brain will prioritize deep sleep first, suggesting that deep sleep is the most essential stage, at least in the short term. Dreams have fascinated humans for millennia. Ancient cultures believed dreams were messages from gods or spirits. Freud believed dreams revealed unconscious desires and conflicts. Modern neuroscience views dreams as a byproduct of brain activity during sleep, particularly during REM sleep. The exact function of dreaming remains debated. Some researchers think dreams serve no function, that they're merely random neural firing during sleep, like static on an untuned television. Others believe dreams help process emotions, allowing you to work through feelings in the safe context of sleep. Still others propose that dreams help consolidate memories or practice responses to threatening situations. The truth might involve all these functions to some degree. Dreams might not have a single purpose, but rather emerge from multiple processes happening during sleep, each serving its own function. Not all dreaming occurs during REM sleep. You can dream during non-REM sleep too, though non-REM dreams tend to be less vivid, less bizarre, and more thought-like than the hallucinatory narratives of REM dreams. Some people report never dreaming, but this likely reflects difficulty remembering dreams, rather than a true absence of dreaming. If you wake someone during REM sleep, they'll report a dream about 80% of the time. If you wake them during non-REM sleep, they'll report some mental content about 50% of the time. But by morning, most dreams are forgotten. Dream recall fades rapidly unless you make an effort to remember, to write down dreams immediately upon waking. Dreams are ephemeral, existing in that liminal space between consciousness and oblivion, meaningful yet transient, familiar yet strange. The average person spends roughly 2 hours each night dreaming, according to sleep research, though you might not remember any of it. The content of dreams draws from your waking experiences, your memories, your concerns, and preoccupations, but reassembled in novel, often bizarre ways. Dreams can feature people you know, doing things they would never do, places that blend multiple real locations, scenarios that violate physical laws. This creative recombination might be valuable. Some researchers suggest that dreams help the brain make unexpected connections between ideas, potentially facilitating creative problem solving. The story of the chemist Auguste Kekulé discovering the ring structure of benzene after dreaming of a snake biting its own tail is often told, though historians debate its accuracy. Whether true or not, it captures this idea that dreams can inspire creative insights by allowing the brain to explore unusual associations. Sleep deprivation has profound effects on the brain. After just one night without sleep, attention, memory, and reaction time all decline. After several days without sleep, cognitive impairment becomes severe. Hallucinations can occur. Mood disturbances are common. The longest documented case of voluntary sleep deprivation lasted 11 days, achieved by a high school student in 1964 as a science fair project. He experienced mood changes, memory problems, and difficulty concentrating, but recovered fully after sleeping. However, chronic sleep deprivation, even of just 1 or 2 hours per night over weeks or months, accumulates into a significant sleep debt. People adapt to this chronic deprivation, feeling less sleepy over time, but objective measures of performance continue to decline. You can't train yourself to need less sleep. The brain requires sleep for proper function, and there's no way to avoid that biological imperative. Sleep helps the brain clear out metabolic waste products that accumulate during waking activity. Research suggests that during sleep, particularly during deep sleep, cerebrospinal fluid flows through the brain more rapidly, washing away toxins, including proteins like beta amyloid that are associated with Alzheimer's disease. This process, sometimes called the brain's glymphatic system, appears to be more active during sleep than during waking. One hypothesis is that sleep deprivation may be harmful, partly because it prevents this cleaning process, allowing toxic proteins to accumulate. Chronic sleep deprivation has been linked to increased risk of neurodegenerative diseases later in life. Sleep isn't just mental downtime, it's active maintenance time. When the brain repairs itself, clears waste, prepares for the next day of waking activity, consciousness itself remains one of the deepest mysteries in neuroscience. How does the physical activity of neurons give rise to subjective experience, to the feeling of what it's like to be you? This is sometimes called the hard problem of consciousness. We can correlate brain activity with conscious states. We know that the prefrontal cortex and posterior parietal cortex are crucial for conscious awareness. We can track how information flows through the brain, how sensory signals are transformed into perceptions, decisions, and actions. But explaining why any of this feels like something, why there's an experiential quality to it, remains deeply puzzling. Some philosophers and scientists think consciousness will eventually be explained by neuroscience, that once we understand the brain well enough, consciousness will no longer seem mysterious. Others believe consciousness involves something beyond physical processes, something that will never be fully explained by studying neurons and synapses. For now, consciousness remains a profound mystery, a reminder of how much we still don't understand about the brain. The two hemispheres. Your brain is divided into two hemispheres, left and right, separated by a deep fissure. Despite being nearly mirror images anatomically, these hemispheres have somewhat different functions. This functional specialization is called lateralization. Language functions are lateralized to the left hemisphere in about 96% of right-handed people, and roughly 60 to 70% of left-handed people. Damage to the left hemisphere often causes language problems, while damage to the right hemisphere typically doesn't, at least not to the same degree. This lateralization of language was one of the first discoveries in neuropsychology made in the 19th century by examining patients with brain damage. A French physician named Paul Broca identified a region in the left frontal lobe that, when damaged, caused difficulty producing speech, while leaving comprehension relatively intact. A German physician named Karl Wernicke later identified another region in the left temporal lobe where damage caused difficulty understanding language, while leaving speech production fluent, albeit nonsensical. The two hemispheres communicate through the corpus callosum, a thick bundle containing an estimated 200 million or more nerve fibers connecting corresponding regions in each hemisphere. The corpus callosum allows the hemispheres to share information, to coordinate activity, to work together as a unified brain. Some people are born without a corpus callosum, a condition called agenesis of the corpus callosum, and many live relatively normal lives, suggesting that the brain can adapt, developing other pathways for interhemispheric communication. In rare cases, the corpus callosum is surgically severed to treat severe epilepsy. These split brain patients provided neuroscientists with a unique opportunity to study hemispheric function, revealing just how different the two hemispheres can be in their capabilities and processing styles. Studies of split brain patients revealed fascinating details about hemispheric specialization. In these patients, information presented to the left visual field goes only to the right hemisphere, and information presented to the right visual field goes only to the left hemisphere because the connecting fibers have been severed. If you show a split brain patient an object in their left visual field, activating the right hemisphere, they can't name it because language is in the left hemisphere and the hemispheres can't communicate, but they can draw it with their left hand, which is controlled by the right hemisphere. If you show the same object to their right visual field, activating the left hemisphere, they can easily name it but might have trouble drawing it because the left hemisphere is less skilled at spatial tasks. Each hemisphere has access to only half of the visual world and to its own specialized abilities, creating a peculiar dissociation between knowing and being able to express what you know. The right hemisphere specializes in spatial processing, facial recognition, and processing the emotional tone of language. When you recognize someone's face, your right hemisphere is working hard. When you navigate through space, remembering how to get somewhere, your right hemisphere is crucial. When you understand the emotional subtext of what someone is saying, the sarcasm or warmth in their voice, that's largely the right hemisphere's doing. Damage to the right hemisphere can cause problems with spatial orientation, difficulty recognizing faces, and trouble understanding the emotional aspects of communication. A person with right hemisphere damage might understand the literal meaning of words, but miss the emotional tone, the context, the subtle social cues that give communication its full richness. Despite these hemispheric differences, the idea that people are left-brained or right-brained is a myth. Some popular psychology books and educational programs claim that left-brained people are logical and analytical, while right-brained people are creative and intuitive. This is an oversimplification to the point of being misleading. Both hemispheres contribute to virtually every cognitive task. Creativity involves both hemispheres. So does logical thinking. While there are real differences in hemispheric function, they're subtle, involving how information is processed rather than what information is processed. Both hemispheres work together constantly, sharing information through the corpus callosum, cooperating to produce unified cognition and behavior. The left hemisphere tends to focus on details, processing information in a sequential, analytical way, attending to the parts of a pattern. The right hemisphere tends to focus on the big picture, processing information holistically, attending to overall patterns and contexts. These complementary processing styles work together, allowing you to see both the forest and the trees. When you look at a face, the left hemisphere might process individual features, noticing the shape of the nose or the color of the eyes, while the right hemisphere processes the face as a whole, recognizing the person and their emotional expression. Neither approach is better. Both are necessary for full understanding. Handedness is related to brain lateralization, though not as simply as you might think. About 90% of people are right-handed, and the vast majority of right-handed people have language lateralized to the left hemisphere. But left-handedness is more variable. About 60 to 70% of left-handers have language in the left hemisphere, just like right-handers. Another 30 to 40% have language either in the right hemisphere or distributed across both hemispheres. This variability makes left-handers interesting to neuroscientists. They show greater diversity in brain organization, suggesting that lateralization isn't rigidly determined, but can vary from person to person. Handedness itself is controlled by motor areas in the hemisphere opposite to the preferred hand, so right-handers have dominant left motor cortex, and vice versa. The two hemispheres don't just process different types of information, they also seem to interact differently with each other. The left hemisphere tends to make strong, within-hemisphere connections, allowing for rapid, focused processing. The right hemisphere makes stronger between-hemisphere connections, allowing for more integrative, context-sensitive processing. These different connectivity patterns might explain some hemispheric differences. The left hemisphere's dense, local connectivity supports the rapid, sequential processing needed for language. The right hemisphere's broader connectivity supports the integration needed for spatial processing and understanding emotional context. The brain's architecture reflects its function, with structural differences between hemispheres supporting their different processing styles. Some research suggests that the two hemispheres might have slightly different ways of dealing with the world. The left hemisphere seems more optimistic, more likely to see the positive, to stick with current theories and explanations even when evidence suggests they might be wrong. The right hemisphere seems more attuned to negative information, to threat, to things that don't fit expectations. This might not be a bug, but a feature. The left hemisphere's optimistic stance helps maintain stable beliefs and behaviors, allowing you to function without constantly questioning everything. The right hemisphere's skeptical stance helps detect problems, notice when something's wrong, alert you to potential dangers. Together, they balance confidence with caution, optimism with realism, allowing for both stable functioning and adaptive change. Perhaps most importantly, the two hemispheres give the brain a kind of backup system. If one hemisphere is damaged, the other can sometimes compensate, taking over functions that would normally be localized to the damaged side. This is especially true in children, whose brains are more plastic. Children who have an entire hemisphere removed surgically, a procedure called hemispherectomy, sometimes used to treat severe epilepsy, can recover remarkably well. Language and other functions gradually reorganize in the remaining hemisphere. In adults, this kind of dramatic reorganization is less common, but some recovery is still possible through rehabilitation and neuroplasticity. The fact that one hemisphere can sometimes substitute for the other suggests that hemispheric specialization, while real, isn't absolute. The brain is flexible, adaptive, capable of reorganizing itself when necessary to maintain function. Sensory processing and perception. Everything you know about the world comes through your senses, filtered and interpreted by your brain. Vision, hearing, touch, taste, smell, all begin with specialized receptors that convert physical energy into neural signals. But sensory processing is far more than passive reception. Your brain actively constructs perceptions using incoming sensory information combined with expectations, memories, and context to create your subjective experience of the world. What you perceive isn't a direct readout of reality. It's a carefully constructed interpretation shaped by your brain's predictions and assumptions about what's likely to be out there. Vision is the dominant sense for most humans. About 30% of your cortex is devoted to visual processing, far more than any other sense. Visual processing begins in the retina, where photoreceptors convert light into neural signals. These signals travel through the optic nerve to the thalamus, then to primary visual cortex at the back of your brain. From there, information flows through multiple specialized visual areas, some processing color, others motion, others shapes and objects, still others faces and places. This parallel processing allows the brain to analyze different aspects of the visual scene simultaneously, then integrate them into a unified perception. What seems like effortless seeing actually involves dozens of brain areas working together, each contributing a piece to the final perceptual experience. Your brain fills in missing information in your visual field all the time. You have a blind spot in each eye where the optic nerve exits the retina, a spot where there are no photoreceptors. But you never notice a dark spot in your vision because your brain fills in the gap, extrapolating from surrounding areas, making up details based on what's likely to be there. This filling in happens continuously throughout your visual field. The center of your vision, where you're looking directly, has high resolution, but peripheral vision is much less detailed. Your brain fills in the periphery based on past experience and expectations, creating the illusion of a seamlessly detailed visual world. You can demonstrate this yourself. Hold your hand out to the side while looking straight ahead. You can detect motion and rough shapes in your peripheral vision, but you can't read fine details without moving your eyes to look directly at your hand. Color, doesn't exist in the physical world. Objects reflect certain wavelengths of light and absorb others, but the light itself has no color. Color is a creation of your brain, a way of representing information about wavelengths of light in a form that's useful for behavior. Your retina contains 3 types of cone photoreceptors, each sensitive to different ranges of wavelengths. Your brain compares the signals from these 3 cone types and constructs the experience of color. This is why people with color blindness, who have defective or missing cone types, perceive colors differently. It's also why colors can look different under different lighting conditions. Your brain adjusts color perception based on context, trying to maintain stable color appearance despite changes in illumination. The dress that went viral on the internet, appearing blue and black to some people and white and gold to others, highlighted how the brain makes assumptions about lighting when interpreting color, and how those assumptions can vary from person to person. Hearing begins with vibrations in the air traveling through your ear canal, causing your eardrum to vibrate, which in turn causes tiny bones in your middle ear to vibrate, which causes fluid in your cochlea to move, which finally causes hair cells, the actual sensory receptors, to fire. These hair cells convert the mechanical vibration into neural signals that travel to auditory cortex in your temporal lobe. But the brain doesn't just passively receive sounds. It actively constructs auditory perceptions, separating sounds from different sources, filling in missing information, suppressing irrelevant background noise. When you listen to someone speaking in a noisy room, your brain is working hard to separate their voice from the cacophony of other sounds, using cues like location, pitch, and speech patterns to isolate the relevant auditory stream. This is called the cocktail party effect, and it demonstrates how much active processing is involved in seemingly effortless hearing. Touch involves multiple types of receptors in your skin, each sensitive to different types of stimulation. Some respond to light touch, others to pressure, others to vibration, others to pain, others to temperature. Signals from these receptors travel up your spinal cord to your brain, where they're processed in somatosensory cortex. Different body parts are represented by different amounts of somatosensory cortex, with more sensitive areas like your hands and lips getting more cortical territory than less sensitive areas like your back. This is reflected in the sensory homunculus, a distorted map of the body drawn on somatosensory cortex, where hands and lips are enormous and torso is tiny. The homunculus reflects the importance of different body parts for interacting with the world. Your hands need fine, discriminative touch to manipulate objects. Your lips help with both eating and speaking. These high priority areas get more neural resources. Pain is a special type of sensation, essential for survival but unpleasant by design. Pain signals travel from nociceptors, pain receptors throughout your body, through specialized pathways in your spinal cord to your brain. But pain isn't just a simple sensation. It's a complex experience involving sensory, emotional, and cognitive components. The sensory discriminative component tells you where the pain is and what type of pain it is. The affective motivational component makes pain unpleasant and motivates you to avoid it. The cognitive evaluative component interprets pain based on context and expectations. This is why pain can be modulated by attention, emotion, and beliefs. Placebo treatments can reduce pain through expectation effects. Distraction can make pain less noticeable. Anxiety can amplify pain. The brain constructs pain experience from multiple sources of information, not just sensory signals from damaged tissue. Taste and smell are chemical senses, detecting molecules in the environment. Taste involves receptors on your tongue, sensitive to 5 basic taste qualities: sweet, sour, salty, bitter, and umami, a savory taste associated with amino acids. But flavor, the experience you have when eating, involves much more than taste. It combines taste with smell, with texture, with temperature, with expectations and memories. Much of what we think of as taste is actually smell. When you have a cold and your nose is stuffed up, food seems bland because you're missing the olfactory component of flavor. Smell is a unique sense in that olfactory information doesn't pass through the thalamus on its way to cortex. It goes directly to olfactory cortex, then to regions involved in emotion and memory. This direct connection might explain why smells can trigger vivid memories and strong emotions, why the scent of cookies baking can transport you back to your grandmother's kitchen decades ago. Perception is fundamentally predictive. Your brain doesn't just passively receive sensory information and respond to it. Instead, it constantly generates predictions about what it expects to sense next, based on past experience and current context. These predictions are compared with incoming sensory information. When predictions match sensory input, perception proceeds smoothly, effortlessly. When predictions and sensory input mismatch, the brain updates its model of the world. This predictive processing explains many perceptual phenomena. Visual illusions often result from the brain making incorrect predictions about sensory input. Surprise, that feeling when something unexpected happens, reflects a prediction error, a mismatch between what your brain expected and what actually occurred. Your perceptual experience is always a blend of bottom-up sensory information and top-down predictions. You never perceive the world directly. You always perceive an interpreted, predicted version constructed by your brain. The brain's predictive nature means that context profoundly shapes perception. The same physical stimulus can be perceived differently depending on what surrounds it, what came before it, what you expect to encounter. In vision, this is called contextual modulation. A gray square looks lighter when surrounded by black than when surrounded by white, even though the gray square itself is identical. Your brain interprets the gray based on its context, adjusting perception to account for apparent lighting conditions. Similar context effects occur in all senses. A sound seems louder in silence than in noise. A touch feels more intense when unexpected than when anticipated. The brain is constantly using context to interpret sensory input, ensuring that perception reflects not just the immediate stimulus, but its meaning within the broader situation. Your sensory systems adapt to sustained stimulation, a phenomenon called sensory adaptation. When you first step into a cold pool, the water feels shockingly cold, but after a few minutes, you adapt, and the water feels comfortable. Your temperature receptors have adjusted their sensitivity, becoming less responsive to the sustained cold stimulus. This adaptation occurs in all sensory systems. Your eyes adapt to dim light, becoming more sensitive, allowing you to see better in darkness. Your sense of smell adapts to sustained odors, which is why you stop noticing your own perfume or the smell of your home after a while. Adaptation allows sensory systems to remain sensitive to changes in stimulation, while preventing sustained, unchanging stimuli from overwhelming perception. It ensures that your senses respond most strongly to new information, to changes that might signal something important, rather than wasting resources signaling constant, unchanging aspects of your environment. Attention powerfully modulates sensory processing. When you attend to something, neural responses to that stimulus increase throughout sensory cortex. When you ignore something, even though it's physically present, neural responses decrease. This attentional modulation begins surprisingly early in sensory processing, affecting primary sensory areas, not just higher-level association areas. Attention acts like a spotlight, enhancing processing of attended information while suppressing unattended information. This selectivity is essential because your sensory organs take in far more information than your brain can fully process. Without attention's filtering function, you'd be overwhelmed by sensory input, unable to focus on what matters. Attention allows you to prioritize, to allocate your limited processing resources to the most relevant information. The phenomenon of change blindness reveals how much of the world you don't actually perceive, despite the illusion of seeing everything. When viewing a complex scene, if something changes during a brief interruption, like an eye blink or cut in a video, people often fail to notice even dramatic changes, like a person being replaced with someone wearing completely different clothes. This happens because your brain doesn't maintain a detailed representation of everything in view. Instead, it represents only what you're attending to, relying on the fact that you can shift attention to any part of the scene if needed. Change blindness demonstrates that perception is selective, constructed, incomplete. What feels like a rich, detailed perception of your entire visual field is partly an illusion. Much of the detail exists in the world, not in your brain's representation, and your brain relies on being able to look at things again, if needed, rather than maintaining complete internal representations. Brain power and performance. The human brain generates approximately 20 watts of electrical power, about enough to light a dim bulb. This might not sound like much, but consider what it accomplishes with that modest power budget. Every thought, every sensation, every memory, every decision, all running on the energy equivalent of a nightlight. Compare this to modern computers, which might use hundreds of watts to perform tasks that brains do effortlessly, like recognizing faces or understanding natural language. The brain achieves remarkable computational efficiency through massive parallelism, with billions of neurons working simultaneously, and through analog rather than digital processing. Neurons don't just send binary on-off signals. They send graded signals, varying in strength and timing, carrying more information per signal than a simple binary bit. Information travels through your brain at varying speeds, depending on the type of nerve fiber. The fastest signals, traveling down heavily myelinated nerve fibers, can reach speeds up to about 270 miles per hour. But not all neural signals travel that fast. Unmyelinated fibers conduct much more slowly, sometimes less than 2 miles per hour. The brain uses different speeds for different purposes. Rapid signals support quick reactions, like pulling your hand away from something hot. Slower signals might suffice for more leisurely processes, like gradually adjusting your arousal level or modulating background mood. This diversity of conduction speeds allows the brain to match signal speed to functional requirements, using faster, more metabolically expensive conduction only when necessary. A single neuron can fire about 200 times per second at maximum, though most fire much less frequently on average. This might seem slow compared to computer processors that operate at billions of cycles per second. But neurons compensate for their relatively slow firing rates through massive parallelism. While a computer processor might execute instructions one at a time, billions of neurons in your brain fire simultaneously, processing information in parallel across vast networks. This parallel architecture allows the brain to perform certain tasks, like pattern recognition, much faster than any computer, despite individual neurons being millions of times slower than transistors. The brain uses about 20% of the oxygen your body takes in, despite representing only 2% of body weight. This high oxygen demand reflects the brain's intense metabolic activity. Neurons require constant energy to maintain their resting potential, to generate action potentials, to synthesize neurotransmitters, to repair and maintain cellular machinery. If blood flow to the brain is interrupted, even for a few seconds, you lose consciousness. After 4 to 6 minutes without oxygen, brain cells begin to die. This extreme oxygen dependence makes stroke, which interrupts blood flow to parts of the brain, such a devastating condition. Without blood flow, neurons can't generate energy, can't maintain their ionic balance, can't function. Rapid restoration of blood flow is crucial for minimizing brain damage after stroke. Your brain constantly updates itself, refining its wiring through experience. This process doesn't stop in childhood or adolescence. It continues throughout life, though the rate and extent of change decrease with age. Every new memory, every new skill, every new experience leaves a trace in your brain's structure, strengthening some synapses, weakening others, occasionally creating entirely new connections. This ongoing plasticity means your brain is never static, never fixed. It's a dynamic organ, continuously adapting to your experiences, continuously learning, continuously changing. The person you are today has a subtly different brain than the person you were yesterday, reshaped by yesterday's experiences, prepared in new ways for tomorrow's challenges. You can only consciously process a tiny fraction of the information your brain handles. Most of what your brain does operates beneath awareness, automatically, unconsciously. You're not aware of your brain maintaining your heartbeat, regulating your breathing, adjusting your blood pressure, coordinating your muscles when you walk. You're not aware of the vast amount of sensory processing happening in early visual areas, in auditory cortex, in somatosensory areas. Only a small spotlight of consciousness illuminates part of the brain's activity at any moment, the part you're attending to, the part relevant to your current goals and concerns. The unconscious work of the brain is not inferior to conscious processing. In many ways, it's more efficient, more reliable, more sophisticated than conscious thought. Consciousness may be impressive, but most of what makes you work happens in the shadows, outside awareness, managed by neural systems that have been refined over millions of years of evolution. Different brain regions operate at different speeds. Some areas respond to sensory input within a few dozen milliseconds. Others, particularly those involved in higher order cognition, like the prefrontal cortex, might take hundreds of milliseconds to process information. This temporal hierarchy reflects the flow of information through the brain, from fast sensory processing to slower, more abstract, more integrative processing. By the time you become consciously aware of something, your brain has already done substantial processing, already filtered and interpreted the information, already initiated some responses. Consciousness arrives late to the party, catching up with processes that began milliseconds or even seconds earlier. The brain's performance varies with state. When you're alert and engaged, your brain performs differently than when you're tired, stressed, or distracted. Stress, for instance, enhances some types of cognitive function, like vigilance and fast reaction to threat, but impairs others, like working memory and cognitive flexibility. This makes sense from an evolutionary perspective. When facing immediate danger, you need quick reflexes and heightened awareness, not deep contemplation. But this same stress response, when chronic, can impair learning, memory, and decision-making. The brain state, modulated by factors like sleep, stress, hunger, hydration, and overall health, profoundly influences cognitive performance. Optimizing these state variables is often as important for cognitive performance as directly training cognitive skills. The aging brain shows both decline and preservation. Some abilities, particularly those involving speed of processing and working memory, tend to decline with age. Older adults often process information more slowly, struggle more with multitasking, have more trouble with tasks requiring rapid decisions. But other abilities, particularly those involving accumulated knowledge and expertise, remain stable or even improve with age. Vocabulary, general knowledge, wisdom about human nature and social situations. These often continue developing well into old age. The aging brain also becomes somewhat less plastic, less able to learn new things quickly. But it compensates with vast stores of knowledge and well-practiced skills. With age comes trade-offs. Some loss of speed and flexibility, some gain in knowledge and expertise. Mental exercise seems to benefit the brain, just as physical exercise benefits the body. Challenging your brain with novel activities, learning new skills, engaging in complex cognitive tasks, all appear to promote cognitive health. Conversely, cognitive decline accelerates with disuse. This doesn't mean doing crossword puzzles will prevent dementia. The relationship between cognitive activity and brain health is more nuanced. What matters is engaging in activities that are genuinely challenging and novel, that force your brain to develop new capabilities rather than simply exercising existing skills. Learning a new language, mastering a musical instrument, taking up a complex hobby. These kinds of activities that push your brain outside its comfort zone seem most beneficial for maintaining cognitive function with age. The brain's mysteries and future. Consciousness remains one of neuroscience's deepest mysteries. We can identify brain regions that seem necessary for consciousness, can track how information flows through the brain, can correlate specific patterns of brain activity with specific conscious experiences. But we still can't explain why any of this feels like something, why there's an inner, subjective quality to consciousness. Philosopher David Chalmers called this the hard problem of consciousness, distinguishing it from the easy problems, like explaining how the brain processes information or controls behavior. The hard problem asks why processing information feels like anything at all. Why doesn't it all just happen in the dark, without any accompanying experience? This question remains unanswered, perhaps unanswerable by current scientific methods, perhaps pointing to something fundamental we don't yet understand about the relationship between physical processes and subjective experience. We still don't fully understand how memories are stored. We know that memories involve changes at synapses, strengthening of connections between neurons. We know that different types of memory depend on different brain structures. We know that memory consolidation happens during sleep. But the neural code for memory, the way information is actually represented in patterns of synaptic strengths and neural activity, remains largely mysterious. How does a specific memory, like your first day of school, exist in your brain? What pattern of synapses encodes that particular experience? How does that pattern remain stable over decades, while the molecules making up your neurons are constantly being replaced? Memory has been called both the brain's most important function and its least understood one. The neural basis of creativity is poorly understood. What happens in your brain when you have a creative insight, when you suddenly see a solution to a problem that was eluding you, when you come up with a novel idea? Some research suggests that creativity may involve reduced activity in prefrontal regions that normally constrain thinking, allowing more free association, more unusual connections between ideas. Other research emphasizes interactions between brain networks, particularly between the default mode network, active during mind wandering, and the executive control network, active during focused attention. Creativity might emerge from a particular balance between these networks, between letting thoughts flow freely and evaluating those thoughts critically. But we're far from a complete understanding of creativity's neural basis. The placebo effect demonstrates the brain's remarkable capacity to influence bodily function through belief and expectation. When people are given an inactive treatment but believe it's an active medication, they often experience real improvements in symptoms. This isn't just imagination. Placebo treatments can reduce pain, improve mood, even affect immune function and hormone levels. Brain imaging studies have shown that placebos activate many of the same brain regions as active medications. The expectation of relief triggers changes in brain activity that produce actual relief. This reveals how much the brain's beliefs and predictions influence not just perception, but actual physiological states. Understanding placebo effects could revolutionize medicine, allowing us to harness the brain's healing capacities more effectively. Some people have synesthesia, a condition where stimulation of one sense triggers experiences in another sense. For synesthetes, sounds might evoke colors, numbers might have distinct personalities, words might have tastes. Synesthesia appears to result from unusual crosstalk between sensory areas of the brain, perhaps from extra connections that normally get pruned away during development, but persist in synesthetes. Studying synesthesia provides insights into how the brain normally keeps different senses separate and how perceptual experience is constructed. It also hints at the brain's potential for unusual forms of perception, for sensory experiences quite different from those most people have. The default mode network, a set of brain regions that becomes active when you're not focused on any specific task, has emerged as one of the most interesting discoveries in recent neuroscience. This network activates during mind-wandering, daydreaming, thinking about yourself and others, remembering the past, imagining the future. Initially dismissed as mere resting state activity, the default mode network is now recognized as crucial for self-reflection, social cognition, and memory consolidation. Its activity anti-correlates with brain networks involved in focused attention to external tasks. When you're concentrating on something, the default mode network quiets down. When your attention wanders, it lights up. This network might be essential for the ongoing narrative of your life, for maintaining your sense of self across time and across different contexts. Artificial intelligence and neuroscience are increasingly influencing each other. Insights from neuroscience inspire new AI architectures, while AI systems provide models that neuroscientists can use to test theories about how the brain works. Deep learning networks, though vastly simpler than real brains, capture some principles of neural information processing, learning representations of information through multiple layers of processing units. Studying these networks helps neuroscientists understand how biological neural networks might learn and represent information. Meanwhile, understanding the brain's remarkable efficiency and capabilities inspires computer scientists to develop new AI approaches, particularly for problems like pattern recognition, sensory processing, and motor control, where brains still vastly outperform artificial systems. The connectomy, a complete map of all connections in the brain, is a major goal of modern neuroscience. We now have complete connectomies for small organisms, like the roundworm C. elegans, with only 300 neurons. For larger brains, including the human brain with its 86 billion neurons, mapping every connection remains a monumental challenge. But progress is being made. New imaging technologies allow neuroscientists to trace individual neurons across large volumes of brain tissue. Machine learning helps automate the painstaking work of reconstructing neural circuits from imaging data. As connectomics advances, it promises to reveal principles of brain organization, to show how neural circuits compute, to identify changes in connectivity associated with disease. A complete human connectomy remains years or decades away, but partial connectomies of specific brain regions are already providing valuable insights. Brain-computer interfaces, devices that allow direct communication between the brain and external devices, are moving from science fiction to reality. Simple interfaces that decode neural signals associated with intended movements already allow paralyzed individuals to control robotic arms or computer cursors with their thoughts. More sophisticated interfaces might someday allow direct neural control of prosthetic limbs with the full richness of natural movement, or even enable communication for people who have lost the ability to speak. These technologies raise profound questions. If you can control a prosthetic limb with your brain, is it part of you? If your thoughts can be decoded by a computer, what does that mean for privacy? As brain-computer interfaces advance, they will challenge our understanding of the boundaries between mind and machine, between self and tool. Mental health conditions involve alterations in brain structure and function, but we still don't fully understand the neural basis of most psychiatric disorders. Depression, anxiety, schizophrenia, bipolar disorder, autism. Each involves characteristic patterns of brain activity and connectivity, yet the mechanisms remain incompletely understood. This is partly because mental health is multifaceted, involving not just neurotransmitter levels, but also neural circuits, hormones, immune function, genetics, and environmental factors, all interacting in complex ways. Future treatments might be more targeted, aimed at specific dysfunctional circuits or molecular mechanisms, personalized to each individual's unique brain characteristics. We're beginning to see the outlines of precision psychiatry, where treatments are matched to individuals based on brain imaging, genetic profiles, and other biomarkers. The brain's relationship with the body is bidirectional and profound. Your brain controls your body, but your body also influences your brain. The gut microbiome, the trillions of bacteria living in your digestive system, communicates with your brain through the vagus nerve and through the bloodstream, affecting mood, stress, response, even cognition. Exercise changes your brain, promoting neurogenesis, enhancing memory, reducing depression. Diet affects brain function, with deficiencies in certain nutrients impairing cognition and mood. Chronic inflammation anywhere in the body can affect the brain, contributing to depression and cognitive decline. The brain isn't an isolated information processor. It's embedded in a body, constantly influenced by the body's state, constantly sending signals back to maintain that state. Understanding the brain requires understanding its connections to the rest of the body, the ways that brain and body form an integrated system. Neuroscience is on the cusp of revolutionary advances. New technologies for recording from many neurons simultaneously, for manipulating specific cell types with light or chemicals, for imaging the living brain with ever greater detail, all promise to accelerate our understanding. Large-scale collaborative projects are mapping the brain at multiple scales, from molecules to cells to circuits to whole brain networks. Computational models are becoming more sophisticated, able to capture more of the brain's complexity. As these technologies and approaches mature, neuroscience will answer questions that currently seem mysterious. How do networks of neurons produce coherent behavior? How is information represented and transformed as it flows through neural circuits? How do brains learn, remember, make decisions? These questions, unanswerable just decades ago, are yielding to modern neuroscience's increasingly powerful tools and insights. The brain remains the most complex object we know of in the universe. 3 pounds of tissue containing 86 billion neurons, each connected to thousands of others, creating networks of staggering intricacy. From these networks emerge thought, feeling, consciousness itself. After centuries of study, we've learned much about how the brain works, but vast territories remain unexplored. Countless mysteries await discovery. Each answer raises new questions. Each discovery reveals new depths of complexity. The brain studying itself is perhaps the ultimate scientific challenge, a journey of understanding that may never fully reach its destination, but that reveals wonders at every step. And in a way, that's fitting. The brain is not just an object to be studied. It's the source of the curiosity driving the study, the tool being used to understand itself, the mystery investigating mystery, a quiet conclusion. And so we've reached the end of our journey through the brain's landscape. 100 facts about the 3-pound universe residing in your skull. We've wandered through forests of neurons and oceans of neurotransmitters. We've explored the architecture of memory and the chemistry of emotion. We've watched the brain develop and change, sleep and dream, divide its labor between hemispheres, construct the world of perception from scattered sensory signals. We've seen how this remarkable organ generates thought from electricity, experience from molecular interactions, consciousness from the coordinated activity of billions of cells. Every fact we've encountered tonight is really an invitation to wonder. The brain is not just a scientific puzzle to be solved, but a source of endless fascination. It's what makes you, you. Your memories, your emotions, your thoughts, your personality, all arise from patterns of activity in your brain. Understanding the brain is a way of understanding yourself, understanding the physical basis of your inner life, the material substrate of consciousness and identity. As you drift towards sleep now, your brain continues its work. It doesn't rest when you rest. It's processing the day's experiences, consolidating memories, clearing waste, preparing for tomorrow. In a few hours, you'll cycle through stages of sleep, from light to deep to REM and back again, your brain moving through different states of activity, each serving its purpose. You might dream, experiencing a world constructed entirely within your skull, disconnected from external reality, yet feeling vivid and real. Your sleeping brain will keep your heart beating, your lungs breathing, maintaining all the unconscious functions that keep you alive while consciousness fades away. And tomorrow, you'll wake. Your brain will return to its waking state. Consciousness will return, and you'll step back into the world. But perhaps now, with all you've learned tonight, you'll carry a new appreciation for what's happening beneath your awareness, for the billions of neurons firing in coordination, the neurotransmitters flowing across synapses, the networks lighting up in response to every sight and sound and thought. You carry a universe within you, more complex than galaxies, more mysterious than the deepest ocean. If tonight's journey has brought you some peace, some wonder, some new appreciation for this remarkable brain we all share, I hope you'll return. There's always more to explore, always more mysteries waiting to be uncovered, always another night of quiet learning. Consider subscribing to the channel so you'll know when new journeys become available. And if you've enjoyed this exploration, perhaps share it with someone who might appreciate wandering through the landscape of neuroscience as they drift towards sleep. For now though, rest. Let your thoughts settle like snow. Let your breathing slow. Let consciousness fade gently away. Your brain will take care of the rest, working through the night, preparing for tomorrow's adventures. Sleep well and dream of neurons firing in the darkness, creating the light of thought. Good night.